Mr. Chairman, ladies and gentlemen, great pleasure to be here. So I am a professor of chemistry and I run a department of surgery and cancer. In fact, it's one of the largest clinical departments in Europe, as a matter of fact. There's nearly 1,000 people in the department spread over 6 campuses, which poses interesting management as well as scientific challenges, plus the fact that surgeons are alpha male sociopaths, and I've got 300 of them. So one of the reasons that things are complicated and things fail, therapies, drugs, drug companies, is because life is basically very, very complicated. So as we go forward and try to make use of all these new technologies that are becoming available now, we have to use them to understand human complexity at its very root, because almost all those aspects of complexity, and from that I really mean things like those gene-environment interactions that have already been mentioned, is the resultant of who you are and what likelihood you'll get, how likely you are to get a disease and how likely a therapy is going to interact with you. So I'm going to start by giving some comments about human complexity and the extent of it. Then I'm going to talk about how the changing needs of humanity, increasing population, changes in demographics of disease, etc., are going to impose new healthcare demands as the 21st century goes forward, and how we have to embrace complexity and use technology in the right way to address those. I'll tell you about the technologies available for pulling out and dissecting complexity, and then finally I'm going to tell you about how we try and apply those technologies actually in the real world in clinical environment. So one of the things we're trying to do is to prepare for precision medicine, which has already been mentioned by Larry earlier on. And precision medicine is really about trying to take the fuzziness out of therapy by understanding great depth of biology. But as we uncover more and more information about our complexity, and as we can apply that information effectively to develop new therapies, it will actually change the way that we do healthcare management, it changes the way that we do education, it changes the way that we do research. So the stuff we're talking about today and the next couple of days is going to actually deeply impact on the way that society runs in the future if we're successful in our tasks. So the two big challenges in 21st century medicine are the public and the personal. So personalised healthcare, you want to get the ideal treatment for the patient based on some deep knowledge of the biology. That's typically metrics of genes, proteins, metabolites, and potentially environmental exposures, interactions. And exactly those same interactions create the population phenotype, which affects the likelihood of diseases evolving in the population and the disease risk factors that evolve through changing lifestyle. So the metrics actually can be the same both for individuals and for populations. But also what we want to try and do is make measurements on populations and pull out biomarker information which we can then reapply at the personalised healthcare level. So can we make new discoveries from large numbers of people that we can help with the individual? And the ways that we try and make these measurements, these new metrics and new models, comes really from systems biology, which is going to increasingly dominate the way we think about medicine in the 21st century. And there's two basic sorts of systems biology. The so-called bottom-up, where the system is really the cell, so it could be a yeast, it could be a hepatocyte or something like that, and the system is the genes, the proteins, the metabolites, and how they interact in space and time. That actually is pretty complicated in its own right. We're working on cells so we know what's in there, and you can measure most things, so you can have complete measurement and therefore exact mathematical modelling. Now these sorts of bottom-up systems biology approaches, about 95% of all the world's systems biology, it's fantastically good for writing papers in Nature and Science. It's actually pretty useless for humans, because humans are much more complicated and don't behave according to the rules of cells. So we're supra-organisms, right? And what that means is we have more than one genome in control of our physiology and biochemistry. So you heard about the 20-odd thousand genes in our genome, but in fact we've got other ones sitting inside us. The gut microbes, for instance, which I'm going to say quite a bit about, they have nearly 4 million genes. That's a lot, and they're deeply involved in regulation of many metabolic processes. So earlier it said, why does a Christmas tree have 7 times as many genes as us? Actually, it really doesn't. It has vastly smaller number of genes because a lot of our genomic responsibility we've actually devolved to our symbiotic organisms that live inside us. But the point about humans is that they're incomplete in terms of what we can measure. We have 500 different cell types in the body, all doing different things in space and time and interacting. We can't sample all that in a human without killing them and dissecting them, which is unethical, I believe. And so we have an incompleteness right? We end up with islands of information which we try and build mathematical bridges between in order to understand the system. And also we're uncontrolled and uncontrollable, and therefore we are doing something different when we're trying to do the analysis. What we're trying to do is take what we can and create hypotheses that we can use bottom-up systems biologies to test. Now, one of the great advances in systems biology is actually the ways of mapping biology into complex spaces. So this is an interesting diagram in that it's a gene map that connects the different genes which are defective in a whole range of different diseases onto one diagram. And it's difficult to read there, but there's sort of bone, connective tissue, cardiovascular, dermatological. Actually, there's nearly every sort of human disease can be put onto one map that's sort of genetic. And what you find is that there are, again, we've got no pointer, Mr. Chairman. We have genes that sort of span between very different diseases. And of course, we train doctors to be a nephrologist, hepatologist, a surgeon, or whatever. We don't train them to think about the systemic connectivity of biology which underpins disease. Now, just because there are genetic connections does not mean to say all diseases are genetic in the accepted Mendelian sense. It means there's a predisposition according to the cards that you're dealt with when you're born. And how you play those in relation to your environment, your life, is what determines whether you're healthy, wealthy, or wise. And if you remember what Crick said about the genome, he says it's the blueprint for life. Okay, that's a very nice way of putting it. But you think about, what does a blueprint tell you? It tells you how to build something. So you've got a blueprint for a nuclear power station. It doesn't tell you anything about atoms, atomic physics, how it works, electricity, radioactivity, waste product, anything like that. So a genome is actually rather like that. It's a set of instructions for building an organism. And if we think of it like that, we won't go too far wrong. Now, there's something that's come out of genetics quite useful and interesting, a so-called genome-wide association study, where large numbers of people have their genes scanned in various different ways, and then you do some statistical association with diseases or disease risk factors. So this diagram is a Venn diagram of overlap between the genes that are in common, or not common, between three different irritable or inflammatory bowel conditions. And you can see that IBD, irritable bowel disease, actually has an overlap with the other two, Crohn's and ulcerative colitis. And given these are gut conditions, it's not surprising that there are genetic connections. But if you just look at the connections between IBD and other diseases, you find there are connections to type 1, type 2 diabetes, there are connections to multiple sclerosis, which is a brain disease, lupus erythematosus, ankylosing spondylosis, which is to do with your bones and things like that. So diseases are much more complicated in the way they work and operate, and from a mechanistic point of view, when we're treating those diseases, we need to understand that in much more detail. Now, there are limitations to how much you can do with genome-wide association studies. So this is from a few years ago in a British newspaper. Doctors find the FAT gene, the FTO, unkindly known as the fatso gene, which basically find that people who've got a couple of those actually tend to weigh a bit more than people who've only got one copy of the FTO gene. Now, the thing is that there's a big study was done, published recently, in Nature Genetics, so it must be true. It's called the so-called GIANT study, where 250,000 people were looked at. You can imagine how expensive that is, right, to do the genomics on that. And they found 32 statistically significant GWAS-associated BMI-linked genes. But if you go to supplemental data table number 17, and look at the R squared, the actual amount of variance explained in the population, it's 1.47% for the lot. Okay, so the real title of this paper, which was something like 18 new variants discovered associated with BMI, it should have been, "It's not genetic, you fool," okay? Because if you sit on your arse eating pork rinds all day, you will get fat, right? Irrespective of what your genes are. And the real roots of this come in childhood. So this is a snapshot, a rather uncomfortable one, of British children. 30% of our children under the age of 16 get drunk at least once a week. Okay? 10% of children in the UK smoke, mainly girls, actually in the age of 10 to 15. It's increasing there. And at least 50% of them do not get adequate exercise. So irrespective of our technology and our brilliance, right, this is a fundamental cultural problem that has to be addressed, because if you have sick children, you get sick adults. So we're preparing for worst-case scenarios in the future. The other thing is that things like obesity, if you just look at the epidemiological data, you can see it cannot be simply genetic. So between 1996 and 2007, Americans put on about 1.5 million tons of fat. So well done there. But your genome didn't change in that time, did it? Colorado is up there as the last sort of blue state. It's still blue in 2010. And you might want to think about that in relation to a conversation that might be between Sir Isaac Newton on gravity, Darwin on evolution and natural selection, and the foot-long wiener and mountains, right? And they are all, those things you don't find fat people climbing mountains and things like that. So there are some, that may or may not be the truth, but you might think about it. There are other things that have changed, however. So our microbes have changed, those little bugs inside us. And in fact, Jeff Gordon and co-workers showed a few years ago that obese people have actually got very abnormal microbes. The Firmicutes to Bacteroides ratio is very different, and you can modulate that with diet over a period of weeks to months. And interestingly, about 15% of all the calories that come in from your diet are actually processed through the microbes. Now, they can change very quickly. They can change in a few days, actually in hours, really, right? So one of the interesting questions is, has our microbiome as a species changed so that we're actually better at pulling calories out of the diet than we used to be? And as I said, there's a lot of these genes, okay? This MetaHit consortium identified 3.3 million. That's just been upgraded in the last month to 4 million genes in the microbiome, right? 4 million. And 100 trillion organisms weighing approximately 1 kilogram in each of you, which turns over every 2 days. And during your lifetime, you will put down the toilet 25 tons of bacteria, which is 5 full-grown elephants. Okay, so in terms of biosynthetic capacity, this massively exceeds the whole of the rest of the human body, and it's only in recent years we've been able to study it. So we've got all these bugs sitting around in us, and all these different genes, and there's actually multiple interacting genomes. The karyome is the host chromosome set, which is about 20,000 genes. There's a mitochondrial set. Mitochondria are really symbiotic bacteria going around about 2 billion years. There's this completely unique to everyone microbiome, which is not only in your gut, where most of them are, but they're on your face, your skin, your fingers, etc. There's the viruses which sit inside the microbes, which also help regulate their biochemistry. And even the epigenetics, right, is modulated by the microbiome, which is a strong source of methylating and acetylating agents. So the bugs are deeply in there. And they evolve through our life as well. You get your first set from mum when you're born, and over the breastfeeding, a few months, you get up to about 700 species by the time you're about 6 months old. And by the time you're 3 years old, you have about 1,000 to maybe 1,500 species. And that ecological development is seeded by what you get at birth and depends on how you breastfeed or whether you get formula fed. So you can see all these extra things which dictate this extra ecology, right, which could be impacting on human health. And associated with dysbiosis, that's an abnormality of the gut microbes, there are a whole series of diseases, which I'll come back to in a minute. And then who's there, right? This is some work done on the stomach. You know, we were classically told that the stomach is sterile because it gets acidic very readily and kills all the bacteria. I mean, that's obviously not true. Helicobacter, Nobel Prizes, things like that, have been associated with ulcers. But there's also really weird and wonderful things in there, like Deinococcus radiodurans. That's actually common in deep-sea volcano vents, right? It lives in dilute sulfuric acid at 150 Celsius. And it's inside you, and we have no idea what it's doing at all. Okay, and how did it get there as well? So all these modern diseases, a lot of non-infectious diseases, have got microbiological associations. So the gastric ulcers, colorectal cancers, whole range of autoimmune diseases, type 2 diabetes, obesity, allergic type things, because the microbes tune your immune system in the first 3 years of life, and even things like autism, where the autistic children have got incredibly bizarre microbial sets, and some of those produce potential neurotoxins during brain development. So you can start to see how those connections work as well. And there's a big diversity of diseases here, so you need to think also about that map of the human interacting genes. And so we've got a microbiological interactome and a human one, and they cross over in very complex ways. Okay, so why is metabolism, right, good for looking at this? Well, basically, metabolism is where genes and environment interact. So your metabolic phenotype and your metabolic profile is determined by exactly the same things that determine all these diseases. And people have been looking at metabolism for a long time in one way or another. And this is from a 16th century urine chart. These are pots of piss with colours, tastes and smells associated with diagnoses and treatment. So this is actually sort of 16th century pattern recognition based on urinary chemistry. Well, most of us don't like tasting urine too much anymore. There are a few people that do that. But we can use modern spectroscopic tools for producing that sort of information. I'll come back to that in a minute. So looking at that in a different way, we've got all these intrinsic human factors, our genes and our various bits and pieces, and they interact and change through life with the environment. And the general interaction things now and the environmental things, they've got a new damn ohm word called exposome, so you can do exposomomics if you are so minded, and all of those things actually interact to create the microbiome as well. So there's all the things that determine your health, your disease likelihood, the microbes, and your metabolism lock in together. And the point is your metabolism is statistically and biologically connected to those things. So if you can measure metabolism, you can start to say something about the other stuff. Well, there's lots of advanced spectroscopic and chemical tools that we can use. The most important ones are NMR spectroscopy and mass spectrometry. And the reason they're important is they can generate information on hundreds or thousands of molecules in effectively one analytical run. So they can generate an enormous amount of information. And there's lots of different sorts of experiment, lots of different mass spec tools available. All of them got different analytical capabilities. I don't have to talk about those now. The point is they actually generate vast amounts of data which tell you about your metabolic profile. By using appropriate combinations of these techniques, you can basically phenotype an individual. So in our department, we actually have a large collection of these. This is a much bigger collection of these sorts of machines than you'd find in almost any chemistry department. So we have 10 high-field NMR machines and I think it's 20-something mass spectrometers for clinical diagnostics and biomarker discovery. This is about a $20 million capital investment in our department. So, if you want to see my urine, right, you can, here it is. This is run on a big NMR machine. It's got about 9,000 resolved lines in it. And basically, it is completely sort of sample preparation free. So you can take a sort of little pot of urine, 1/2 ml or something, stick it in a big magnet, bombard it with radio waves, you get a signal out like this, and it covers an enormous amount of chemistry. So things on the right-hand side are things, methyl groups from organic acids and amino acids. The great big peak in the middle there is from urea. Other things on the other side are actually from gut microbial co-metabolites. So in your urine, you actually get a window on what gut microbes are doing as well. And NMR is a physical tool. It's based on quantum mechanics. I don't need to go into that. And the reason the spectrum looks the way it is because of the chemical composition. And the chemical composition is determined by the biological interactions. And what we want out of it is medical information. So what we want to do mathematically is go from complex spectra like this and do a series of mathematical transforms that take us from physics to chemistry to biochemistry to biology and to medicine to recover biomarker information. Now, one of the reasons that these spectra are so complicated, with thousands and thousands of signals in there, far more complicated than if you list all of the indices of all the biochemistry books in the world, partly the complexity is due to the interactions of these genomes. So we've got our primary metabolome, which is determined by our genes. Every cell type in the body has got its own metabolome, so you've got 500 odd different ones in the body. And at the same time, they interact with the gut microbiome, which is providing a constant afferent supply of molecules synthesized by the bacteria and also processed from the diet. And then we also process things in our liver, etc., and stick them back into the gut through the bile, and the microbes can process those as well. So what we end up with is a series of interactive axes of metabolism and combinatorial metabolism where you get multiple sequential transformations by different genomes. And you can see this at a macroscopic level. On the top left up there is cholic acid made in the liver in gram quantities every day. It's glycine and taurine conjugated to produce the bile acids that are responsible for emulsification of fat in the upper gut, secreted into the bile, and where it does its emulsification. But some of the bugs in the upper gut conjugate and regenerate cholic acid, and that can then be moved across. Oh, where did that come from? Thank you very much. Very good. Can create deoxycholic acid, right, which is a combinatorial metabolite. It requires more than one genome to make it. That's a liver toxin and a colonic carcinogen, and it's made by you in your body. So if your bugs are very good at doing that, you've just changed your disease risk factors with relation to liver disease and also potentially to cancer. And if you look at these molecules actually in detail, you find these bile acids, for instance, are signaling molecules. They're not just passive, they actually have effects on our regulatory biochemistry. So this is from the comparing germ-free animals that were born by caesarean section, rats, with normal rats. And this is looking at the liver gene expression patterns of cholesterol biosynthesis and steroid synthesis. And everything that is in red or in blue is co-regulated by microbial signaling. That's most of cholesterol metabolism, okay? And we think about drugs like statins hit here, and these are cytochrome P450s, which are related to drug metabolism. And now you can see that bugs and drugs might interact also, because they're controlling the same things we're trying to drug. And indeed, we've evolved with bugs. We haven't evolved with drugs. So all the drug metabolizing enzymes are actually designed for dealing with microbial and plant toxins. And so there's bound to be an interaction between those. And if you think about personalized healthcare as being optimization of drug therapy for the individual biology, you now think individual biology has to incorporate microbial variation as well. And so what we have is a whole series of axes of signaling between the gut, the liver, the microbes, and the brain, in fact. And the one we've just looked at is the bile acid nuclear receptor axis. There are at least 8 known axes, and they interact in space and time in very complex ways and develop as you develop. And as I'll show you later, these are all potential drug-targeted or targeting axes. So can you see this working in the general population, any of these axes? Well, let's take an example in molecular epidemiology. So this is where we phenotype large numbers of people from the general population, and we have various information about them, like their diet, their BMI, their blood pressure, and things like that. And so what we've done is what we call a metabolome-wide association, where we take all those metabolic variables and we link them statistically to disease risk factors. We can also do some quite good fun things with this, so we can start producing metabolic maps of the world. So, for instance, what we have at the bottom here is people from South China in red, and then northern China there, the Japanese, etc. Honolulu, which is halfway between Japan and America, is halfway between Japan and America metabolically. UK is somewhere near Baltimore for some reason. And then you've got basically these people from Texas. And if you map on top of that sort of BMI, mortality, cardiovascular risk, and diabetes, it goes with where you are in the world. And if you have a Japanese American, I haven't got a slide here, they map up here, okay? So you might be genetically Japanese, but if you live here, you get all the risk factors of living here, okay? So environment, your diet, and what you do with your genes is more important than what those genes are. I mean, there's quite interesting stuff here as well. I mean, if you can see it, these are all males here from Japan, these are females. So here you've got population separations, not just on the town, but actually more separated based on the sex, okay? That's not true in China, where male-female is located to the city. The reason for that tells you something about lifestyle variation. So in Japan, it's much more uniform, and the differences between men and women are more uniform. So Japanese men, they go to work 12 hours a day, come out about 9 o'clock, go and get drunk, do karaoke very badly, and women stay at home doing origami and stuff and looking after the kids. And so that emerges in terms of what actually the biochemistry is like. So we can take all these fantastic spectra and we can regress them against things like body mass index, and we can find things with fantastically significantly correlated, 10 to minus 13, 10 to minus 10. All the things in red are gut microbial, okay? So your gut microbes and microbial signatures link into risk factors relating to body mass and also diabetes and other things as well. Now, how on earth does this work in terms of microbes regulating populations? Now, it turns out that, in fact, microbial metabolites link quite closely, urinary metabolites from bugs, link quite closely to blood pressure and BMI. And this is just one example. This is the aromatic amine axis. And so here in the distal colon, you've got tyrosine and phenylalanine metabolism. In this direction, you go towards cresol, and then cresol sulfate, which is made by mammalian metabolism of cresol, which is made by bugs in the colon. And here's phenylacetate going to phenylacetylglutamine. These are very strongly inversely correlated with diastolic blood pressure. Okay? If you look on the other side of this seesaw, we've got a conversion here from tyrosine to tyramine. Tyramine is a vasopressor, an extremely potent one. And if you have a monoamine oxidase inhibitor drug and you have a blue cheese, you'll end up in hospital with hypertension, which is produced almost immediately because this is such a strong vasopressor. So here you have a seesaw of biochemical interaction where this is also a gut microbial conversion. Depending on the activities, you can generate pharmacologically active species that affect blood pressure. And this compound here, which is an analogous pathway from phenylalanine, beta-phenylethylamine, is actually a root structure of amphetamine, and it's a potent anorectic agent. So your microbes make things that are very much like drugs, and they hit our biochemistry constantly. And so that was the axis here of the aromatic amine CNS axis. And I could go through all of the other ones. Well, what happens if you stick a knife in it? Okay? And if you stick a knife in it, you're doing bariatric surgery. So bariatric surgery We do a lot of that at Imperial. It's for weight loss, of course. It's been around since the 1930s, as a matter of fact. And what you do is you basically disconnect your ileum from the duodenum and reconnect it to the top of your stomach here. So you've got a very small stomach. So guess what? You won't be able to eat very much. So that explains the weight loss. And you get a big weight loss, actually, over about, very quickly, actually, but especially over 36 months. But the really interesting thing is it cures diabetics the following day. Right. So now people are using metabolic surgery, if not obese, to cure diabetes. And about 80% of people who've got diabetes are cured. And the mechanism is unknown. Furthermore, long-term follow-up shows that colorectal and other cancers are reduced in bariatric surgery. So we're interested in understanding what the basis of this. Well, we've just put a knife through all of those guts axes, and guess what? The microbes don't like that very much. So this was published a couple of years ago in PNAS. These are people who've had this bariatric surgery versus just ordinary fat people. And this is 15 months after the RYGB. And there's a massive change, especially in the gamma proteobacteria. They've increased and they've stayed there. That means their signaling has changed. And we can also do studies in rats. The advantage of rats is you can do sham-operated experiments. You do proper controls, so you can open them up but not do bariatric surgery. And you can also kill the animals at the end and find out what all the systemic biochemistry is like. And you get, in rats, exactly this is 8 weeks after the RYGB, massive overgrowth of the gamma proteobacteria. So it's exactly the same in the rats as in human. You can also do all the biochemistry. And again, total changes in the bile acid metabolism profiles, the conjugated, especially the taurine conjugates, post-RYGB. So the bile acid signaling has been changed as well. Now, I haven't got time to go through all the detail on it, but what we've done is basically created a systems model of bariatric surgery, looking at the metabolic interactions and the microbial changes at every single part of the system in order to create an understanding of the deep mechanistic effects of putting a knife through your guts. And what it turns out is actually that the microbiome is an essential gearbox in the metabolic engine. So the effects of surgery change the bacteria, which changes the signaling, which changes the biochemistry, including things relating to metabolic syndrome, obesity, and also things that impact long-term on carcinogenic cancer risk. So this is all based on rat work. We've got a big study just going to be completed in the next couple of months where we followed 200 people for 2 years, and we're going to start analysing that very soon to see if we can find the fundamental reasons why bariatric surgery will cure 80% of diabetics. Of course, if it cures diabetics, it's biochemical. If it's biochemical, it's druggable. If it's druggable, it's worth billions. So that's a good reason for doing it in its own right. The other thing was one thing we tried thinking about was the actual anti-cancer effects. So one of the things we wondered was whether or not there was a change in the mutagenicity caused by the surgery. So we tried taking some fecal water, crappy water, and we did a sort of mutagenicity test on it. And it was a disastrous experiment. The crappy water killed all the cells, absolutely annihilated them. And so we ended up doing toxicity tests. It turns out crappy water is really toxic, so don't drink the fecal water of bariatric surgery patients, right? So that's a good message to take home. And of course, the colon itself is amazingly resistant to all sorts of toxicity, but dedifferentiated colonic cancers are not. So there's actually the potential for discovery of cytotoxic drugs, even in crappy water. We are trying to figure out what the chemicals are in crappy water that actually kill the cells. So watch this space. Okay, let me try and move on and think about personalised healthcare a little bit. And trying to predict the future. So we came up a couple of years ago the idea of pharmacometabolomics, which is basically the metabolic equivalent of pharmacogenomics, where pre-interventional metabolic profiles are used to try and predict outcomes of interventions based on training and test set models. So the principle is really quite simple. If you've got a whole load of individuals floating around in a metabolic space, the position in the metabolic space is determined by the vector interactions of all of the different things in our lives: genes, environment, diet, bugs, etc. So two people that are sitting very close in their metabolic space are biologically very similar. So you might argue that given if they're exposed to a stressor or like an anti-cancer drug, that they will behave in similar ways. So the question is, are there prognostic clusters within the data? So irrespective of the vector contributions for individuals, if you're in that zone, is that a better place to be when you're given an anti-cancer drug than that zone? And the actual answer is yes, it's true. So we could use these sorts of pre-interventional metabolic profiles to predict the toxicity of drugs in experimental animals, and we did that a few years ago. for hepatotoxic drugs. But we were also able to go on and show it could work for all sorts of things. You can do pharmacokinetics, cancer drug efficacy modeling. I'll come back to that in a minute. This is an interesting one, right? Prediction of drug metabolism based on a pre-interventional urine profile. So we took 100 people, gave them 2 Tylenol tablets, took the urine before and urine after, so we've got the metabolic profile of the individual, and we've got what the outcome is in terms of the metabolic fate of the drug. And that's Tylenol, the structure, and there's its glucuronide, its sulfate, and there's the cysteine conjugates and things like that. And what we can then do is backplot the outcome based on the pre-interventional profile. So here are some stacked NMR spectra from the individuals, and one of the things we looked at was the sulfate to glucuronide ratio, right? So people were either weak sulfators or strong sulfators in terms of what they did with the drug. And if you back color code that onto the spectra, you can see there's a difference between the blue ones and the red ones. Those are gut microbial metabolites. Gut microbes even affect Tylenol metabolism. And there's probably 8,000, 9,000 publications of literature on Tylenol metabolism, but nobody ever noticed the dominant metabolic fate was actually altered by drugs. And you can see what, by microbes. And if you look at the structure of acetaminophen, Tylenol, right, versus cresol, which is made by the bugs in the distal colon, because it's a structural analog, they both require sulfation, okay? And if your bugs are very good at making lots of cresol, okay, you deplete sulfation capacity, so when you're challenged with a drug that requires sulfation, there's not enough sulfate, and you're a weak sulfator. No genetics required, just some chemistry and some bugs. Furthermore, 4-cresol will compete with all drugs that are hydroxylated that require sulfation, or all the hydroxylated metabolites of those drugs. Okay, that's hundreds of drugs will be affected by one microbial gene. Okay? So hence the limitations of the use of human genomics in understanding drug metabolism, even for common things. If we look into this in a little bit more detail, we figure out that we have to have something called a conditional metabolic phenotype. The conditional metabolic phenotype is what you need to understand to explain reactions to compounds or interventions. So there's all the genetic tomfoolery on that side, but there's also the fact that there's the microbes switching on and off pathways, generating compounds which can compete or not compete with metabolism, and that's partly dependent on diet as well. And bugs interact in lots of different ways. They can do competitive metabolic substrate utilization, that's cresol and Tylenol, for instance. They can do primary metabolism of orally administered drugs. You take some pills, the first genomes that those pills meet are not human, they're microbial. And so amphetamines are metabolized by bugs. Digoxin is metabolized by bugs. Lots and lots of compounds, hundreds of compounds. They can also provide compounds which switch on and off, metabolizing enzymes. They can also metabolize our metabolites as well, and they can even change the local pH, changing the ionization state of the drug and therefore the bioavailability. We can ask the question, out of all those bugs inside us, which ones are doing important stuff? And there are ways of solving that by doing the co-analysis of the microbial variation with the metabolic variation. And so we can actually, by doing clone library analysis and integrated transomic statistics, we can find which bugs relate to which metabolic profiles and which metabolic hubs. And those are the ones related to Tylenol, and they relate to a couple of different bugs. So if we now know which bugs do what in metabolism to control drugs, can we think of the microbiome as a target for drugs in its own right? So it's got many, many thousand times more, well, certainly hundreds of times more genes than our genome, so there must be lots of druggable targets targeting those different axes. Do you think that's science fiction? Well, it's not. It's been done already. So this is Wallace et al. In Science just a year or so ago, year and a half ago, published a paper showing this drug CPT11, which causes severe GI toxicity, its toxicity can be reduced massively by drugging the microbial glucuronidase, which actually basically retoxifies the drug once it's been secreted into the bile. So the drug is absorbed, detoxified in the liver by glucuronidation, resecreted in the bile, and the bugs recreate the toxic species. But if you do a QSAR study and drug targeting, you can knock out the enzyme without killing the bacteria, and the toxicity goes away entirely. So there is a series of signaling axes, and every single one of them is a potential drug target. Or drugging, not just drugs, but beyond that. So we can think about probiotics and prebiotics changing the ecology. We can think about drugging specific pathways. We can think about highly specific antibiotics which knock out only one species. You can think about synthetic biology to create new organisms for designer ecologies. Right, this is going forward into the future, obviously, and we do have to understand a lot more of the biochemistry before we can do this. But because the relationships between bugs and us change throughout life, the windows for therapeutic intervention also change. So in early life, you're thinking about preventive therapies, and in later life, you're thinking about corrective therapies, etc. Okay, so that's one side of complexity and how you go about it. I want to just finish off by showing you how you deploy these technologies and how this sort of knowledge is useful when you're actually coming to real clinical scenarios. So let's think about just changing the way the 21st century of medicine is done through what we might call something like patient journey phenotyping. As mentioned, Larry mentioned something like this earlier on. When you go into a hospital, it's a journey. Okay, you go in. Okay, you come in and you're ill. The doctors try to decide what's wrong with you. They do some sort of staging if it's a tumor or something like that. Sometimes they'll do genetics. Then they do some sort of procedure, right? And you'll either get better or worse. You'll be a responder or a non-responder. So what we're doing at Imperial is we're putting all these technologies into every stage of the patient journey to increase biomarker discovery for clinical decision-making. And we're also thinking about longitudinally using the technologies, using pharmacometabolomics to say, if we make a measurement here, can we predict what will happen here and use that information to stratify patients? This is an example, actually, of surgery, using these technologies in surgery and the surgical patient journey. Now, most of you will know that surgery is normally a pretty molecule-free zone. There are people with knives, okay, in there, right? And surgeons generally don't end up liking biochemistry very much. But on the other hand, when you're given technologies that enhance their decision-making, they're usually quite open to it. So I just want to say for just a couple of minutes the sort of things that we're doing now. So we've put the world's first solid-state NMR into surgery. It did that about 2 years ago for almost real-time diagnostics. Then we're doing Moldy imaging of tissue slices as well, and also something called the intelligent knife, which I'll come back into in a minute. But if you just do, see, here's a piece of a tumour versus non-tumour, stick in a solid-state NMR machine, 5 minutes later, you've got a diagnostic profile which is incredibly sensitive and specific, right, which tells you, what it's in there. Now, tumour versus non-tumour is not very interesting. What's really interesting is tumour stage, right? And we've developed tools now that allow you to take an off-tumour biopsy, 10 centimetres away, and that will tell you the tumour stage based on the biochemistry because of the field effect of the tumour. Furthermore, and quite scarily, that biopsy will tell you the 5-year survivorship probability of the individual. And that has ethical implications, right? So if you take a biopsy and say, the model says you're going to die in 2 years, what do you do then? Do you operate or not operate? Over to you guys to discuss, okay? But this is what the new technology is now showing us, completely new insights into diagnostics and effectively on a real time scale. We can do tissue slice biochemistry. So here's a frozen section. You run a laser over it, ablate molecules into a mass spectrometer, and so you can now molecularly map every single molecule that's measurable. This will measure 1,000 molecules a time, and so you can raster image the tissue based on each molecular profile. This happens to be an image of something weighing 599. It's a lysolecithin, and then you can superimpose that into the conventional image and find out where the chemistry is coming from. You can do even fancier things with multivariate imaging. So this is a piece of brain, rat brain, imaged by a whole series of different molecules. But what you've got is actually a chemical index at every point in the map with a topographical location. So if you do some fancy chemical statistics, this is self-organizing mapping, this clusters together all cell types and all tissue areas based on chemistry. And because you know where that is, you can say, well, where does that dot belong? It belongs over here. So you can go cell type by cell type, chemistry by chemistry, and light up different regions of the brain. And this one day will replace histopathology because it can do any combination of chemicals without knowing what, staining up, knowing what you want to start with. The most exciting project is the iKnife. Because surgeons use knives, as you may have noticed. A lot of them they have a huge variety of instruments of torture, actually. It's quite remarkable to see the box. But one of the things they like using a lot, they use some carbon dioxide lasers, but they also use diathermy, right? And as they cut through you, it generates smoke. Okay, and if you're doing keyhole surgery where you've got incisions here, you actually have to have exhaust pipes, otherwise the patient blows up like that, right, with all the smoke. So here's a surgical knife with an exhaust pipe on it. Here's one of our surgeons fooling around with a dead pig and a mass spectrometer, and that is a mass spectrum created in less than a second from the smoke being generated, which is actually the lipidomic profile of that tissue. And there is a mass spectrometer in an operating theatre, which has been ruggedized, covered in a shell, so you can put disinfectant on it, and also it's on a trolley, so you can wheel it in and out. Okay? And we've now got diagnostics that can work on these are lung tumour smokes, where we can tell the difference between metastatic adenocarcinomas and primary adenocarcinomas, and we can even say where the primary was. So the memory of where that tumour came from is carried in its biochemistry, which is expressed in the smoke, which you can measure in real time. And the diagnostic capabilities of different smokes are just unbelievable. Every smoke of everything looks as though it's absolutely diagnostic. So what do you do with this? I mean, you've got all this information. You can't give a mass spectrum to a surgeon. He will stab you, okay? He definitely does not want to do that. So what you've got to think about is if the surgeon's hacking around on a piece of tissue, right, you've got to think about expressing the variants in another form, right? A zone of normality. Eventually, the tissue signaled up into an abnormal region. You say, well, what the hell is that? And you compare it with the database, which will give you the hit. Okay, so we've now got some 25,000 different sorts of tumour smoke on record in our laboratory, which we're using. And then, if you think about that, you can go on one step forward, the intelligent endoscope. Right, so when they stick something up your backside, have a look round, you put a carbon dioxide laser in there, smoke a bit of tissue, you can use chemical diagnosis as well as visual diagnoses. And the other thing is that we've also got robots. So this is the Vinci robot. Robots are better at doing certain sorts of surgery than humans because they don't make mistakes if they're programmed properly. And we have virtual reality and augmented reality surgical suites at Imperial where we can actually tie in the technology and test it before we test it on real people. And when you're doing these virtual and augmented reality types of surgery, you're actually looking at a sort of 70-inch plasma screen anyway. That's what the surgeon's doing, looking at that. So you can start to display data and look how they respond to that and what they pay attention to and what they don't pay attention to. So finally, in the last few minutes, I just want to try and put the whole thing together and think about modelling the patient journey. So, as I said, you can have a longitudinal journey by summing many patients together through their hospital experiences, and you can also have the real-time modelling, which can be used for immediate clinical decision-making. So, in the case of surgery, there's a whole series of unmet medical needs. There's inflammatory bowel disease differentiation, Crohn's versus ulcerative colitis, and the distal colon is often very difficult to do histologically. Cancer resection margins, that's very important for things like the breast, how much to cut out, right? And based on the biochemistry of either biopsies or the smoke, you can actually start making decisions. Gut infarction, tissue viability, this is one that actually scares me. So if we look at some of the gut surgeons, if you've got an actual blocked artery, an umbilical artery, part of your gut will die. You've got an embolism or something like that. And what they do is they cut it out and join it together. The way they choose to cut out the bits is they choose what's blue, right, and join the rest together and stitch that up. If they've chosen something that's non-viable, the tissue will break down in 24 hours. You'll get breakage, septicemia, end up in intensive care. So we've got to be able to do the cutting out better than cutting out the blue bits, in my opinion. And there's longitudinal modelling things to do with looking at post-interventional sepsis likelihood as well. And you can that looks like a very similar diagram, actually. It is. What I've just replaced is the surgical operating table with chemotherapy. So this is a completely scalable and translatable model where any patient journey can be looked at. Here you'd be looking at, can you predict, based on chemical measurements prior to intervention, likelihood of being a good or a bad responder based on a series of patient journeys. And in fact, there are other areas of unmet medical need. There's tumour staging, real-time responder, non-responder, and of course, selection of the therapy. Again, science fiction or not? Well, it's not, because we actually already done this last year. So we've shown that with people with unoperable colonic tumours, you can actually predict the degree of toxicity of certain drugs based on pre-interventional plasma metabolite profiles. So if you find that somebody is associated with a high risk of toxicity, choose a different anti-cancer drug. Okay, that's patient stratification in the real world. And similarly, you can also, based on plasma profiles, predict outcomes of things like breast cancer therapy as well. Intensive care, we have 3 intensive care units in my department, and I can tell you they're bloody expensive, right? So this is a small proportion of the population, but they are very, very expensive. So it costs something like $8,000 or $9,000 a day for the person to be in an intensive care bed, and that's before you start the therapy. That's just the infrastructure around them, okay? The other thing is they have about a 30% chance of mortality within 2 weeks. So you've got very expensive patients who die a lot. So that's not good. And the problem, part of the problem is that they're very complicated and lots of things are happening to them very, very quickly. So the more we can put technologies in that enhance rapid diagnosis and accurate diagnosis so the doctor knows what the correct next step is and it's all to do with understanding the complex patient that this is about, right, then we will be able to save a lot of lives. So true personalised healthcare, very high mortality, morbidity, and expensive as well. Actually, we've done a calculation. If you could shorten the journeys of our patients in our 3 intensive care wards by 10%, so say 9 days instead of 10 days, well, you could buy a new 600 megahertz NMR machine every 3 weeks, okay? So those are expensive machines, right? $1 million a pop, okay? So you work it through, okay? So sometimes technology, right, even though it looks very, very expensive, in comparison with the patient journey, right, can really be very cheap. So one of the things that we're trying to do is actually model different sorts of patient journeys, see how expensive they are, and see what the cost of putting technology in to see whether it's actually practical in the real world. And one of the engines that we have for doing that sort of work is our Biomedical Research Centre, our BRC at Imperial, and we have, this is an engine of translation that connects Faculty of Medicine activities with the so-called clinical program group outputs, actually, who are delivering in our National Health Service. So we have basic research here, and we've got implementation over here, and we have, I think it's $180 million over 5 years for doing all these different types of journeys, and also things like stratified medicine, which cross-cuts the whole lot. And if you look at the stratified medicine program at Imperial, top left up here is the surgical journey, the cancer journey, the critical journeys, other journeys, feeding samples into our analytical centres, analysing them using computational medicine methods, and feeding back information to doctors. And at the same time, looking at the socioeconomic value by pairing up with our business school to model the patient journey costs and whether or not it's worth doing in the long run. So finally, what you need is quality, quality, quality. Those are the challenges here, because you've got to be, if somebody's life is depending on it, you've really got to have validated your methods very well. Your biomarkers got to really work. Yeah. You've got to integrate the data with the gold standards, whatever. That's what doctors use, actually. They're not really gold. They're actually sort of brassish in most cases. But it's what doctors actually use. In the future, the gold standards will be systems biology standards and these sorts of technologies. But we have to pay lip service to what's in existence at the moment and try and augment those rather than simply replacement at this stage. And then we've got to process it in very high speed, right, to feed back information very quickly, and we've got to be able to visualize it in a way that's useful for doctors to make decisions. If they can't visualize the data, they won't want to see spectra, they won't want to see complex mathematical models, they want a decision tree saying, based on this, you need to do one of the following things. It's got to be economically viable, and it's also got to be time-proofed. So anything you do has got to be still going to be workable in 5 years' time, even though the technology changes. And that's actually quite a big challenge, because technology does change quickly. So what you need to be thinking about is critical feature extraction from data, which means it's platform independent. And all along, you have to remember people and life is complicated, and you can screw up really easily. And if you don't do that, we will not be able to deploy these technologies in the real world for systems medicine, for personalised healthcare. So thank you to the people who work with us, our collaborators and our people who give us money, and you for kindly listening. Thank you very much.